This video is about exam technique. It's not showing you how to answer the exam paper because the marking instructions for this exam paper have already been put out. I've put all my answers in here, and most of those agree very well with the marking instructions. But really what we're looking at in this paper is the questions that students found difficult last year in 2025. Because quite often what the SQA do is they ask those questions again the next year, because if students find them difficult in 2025, they want to see if teachers have improved their teaching of those topics in 2026. So one of the things that you really must pay attention to, though, and you'll find this in the marking instructions, this is section M in the marking instructions released by SQA, is some ideas about how you should answer questions. So you must answer an identity, name, give, or state question, just a simple, brief answer. If you have to describe something, you must provide a statement as opposed to simply one word. If you're going to explain something, you must provide a reason for the information given. And comparing, you must talk about the similarities and differences between the topics being examined. Calculating, you must come up with a number. Predicting, you need to come up with a number that's in a range, all right? So generally, it's a number that you're asked for. Predict what the temperature would be that something happens. You've got to be within a range of temperatures. You don't have to get the exact answer. You just need to be within the range that they suggest. There's usually an upper and a lower limit that you can work out from the question. And suggest, this may be a question, a kind of an open-ended question, which could give you any one of 3 or 4 possibilities, will each have a different explanation. So there's one of those questions in here. We'll mention it as we pass. Okay, so there's the 2025 paper. This is a photocopy. And the first question that students struggled with was question number 1A, part 1. The very first question in the paper, and the reason they struggled with this paper, I think 75% of students didn't get this. First of all, you had to put in 4 ticks in this table in order to get the marks. You had to put those 4 ticks in. But the biggest problem in this was vacuole. Now, I think a lot of students ticked vacuole because they weren't sure. I don't think anybody who did the exam was very sure about whether vacuole is present in bacteria or not, because to be perfectly honest, the SQA have never asked this before, so a lot of teachers have kind of not really been clear about where the vacuole is. Also, there's not really terribly good information on what SQA consider to be a typical bacterial cell, and typical plant cell, typical fungal cell. But anyway, outside of that, vacuole is only present in eukaryotic cells. It's a membrane-bound organelle, which bacteria don't have. So there are various other issues with this question that I'm not very happy with SQA. This is a person using a light microscope up here, but you don't see ribosomes in a light microscope. You can't technically see a cell membrane either. You're not going to see plasmids. In fact, you're not going to see inside a bacterial cell with a light microscope. So really, I think the SQA could do better there. But anyway, the question was to ask you what was the difference between a bacterial cell and a fungal cell, and you had to take those 4 things to get your 1 mark. A lot of students didn't get it. Okay, question 1C. Explain why a muscle cell contains a large number of mitochondria. You had to link the activity of a muscle cell. You need a lot of energy to be an active cell, and you need a lot of mitochondria to produce the ATP to do the cell's activities. ATP is the currency of energy that cells use. Muscle cells need ATP to contract. Therefore, mitochondria are present in muscle cells to make that ATP. There you go. ATP is not transported over great distances. It's made in the cell that needs it. It's the glucose that's transported over long distances. Sorry about my coughing, everyone. Okay, question 2. Still riffing on the whole vacuole thing. We've got vacuole cell sap. So cell sap is found in the vacuole. They asked that down here, but most people seem to get that one right. Okay, so here the problem was not being specific. You've got to be specific about the stuff that's moving, where it's moving from, and where it's moving to. If you didn't put those things in really clearly, you didn't get the mark. A lot of students in this particular question just said high to low concentration and didn't get the mark for it. In the other one is that if you're going from low to high concentration, it's going to be active transport. You're going up the concentration gradient. Therefore, you need energy. Therefore, you need active transport. And that's what you had to write in there. Name a process that moves the excess sodium ions out of the unicellular organism. They have to use energy. It's active transport. Okay, everyone got that one okay, but we're going to come back to that diagram. So when we were talking about enzymes, in this, we had this prediction. There was a prediction question here. And if we, as we saw in the graph, at 45 degrees Celsius, there was reasonably high activity. At 55 degrees Celsius, there was almost no activity or very low activity. What would you predict to happen at 75 degrees Celsius? Well, you'd expect to be even lower activity or none at all, right? The activity is going down because the enzyme is gradually being denatured. But if you said denatured, you got it wrong, and that's what a lot of people did. They just put denatured in. But they said, what would happen to the enzyme activity? It wasn't asking what would happen to the enzyme. It was asking what happened to the enzyme activity. So you have to say there's less activity or no activity. Either of those would do. You can say because it's denatured, but you don't need to do that to get the mark. Okay, and 90% of the students, a lot of students, failed to answer this one. Describe how this investigation could be changed to find a more exact optimum temperature of invertase. Well, let's have a look at the experiment, right? They were testing different pHs here and 2 different temperatures, right? We see that activity is quite high at 45 degrees Celsius. So all you had to say for that question was to maybe carry out the experiment again at different temperatures. You could carry out the whole experiment again. You'd still have a pH optimum. You could say carry out the experiment at pH 5 at different temperatures between around 45 degrees Celsius. You don't need to go any higher than 55 degrees Celsius because obviously that's lower. So you're going to be looking for temperatures that are above and below 45 degrees Celsius. And 75% of students failed to answer this one about enzymes. Look up enzymes. They ask about them every year. And just because they asked about it last year doesn't mean it won't be in this year. The enzymes are really important molecules in biology, and they ask about it every year. So make sure you know your active sites from your substrates, all right? Explain why invertase would not speed up this reaction of starch to maltose. Well, invertase is specific to sucrose. Sucrose is its substrate. Starch is not its substrate. So the active site of invertase is not specific to starch, or you could say the active site of invertase is specific to sucrose, not starch. But that's what you had to write to get the mark there. Got to be specific and use the terms. Okay. A lot of people didn't know that a section of DNA that codes for a protein is a gene. That's what a gene is. It's just a part of the DNA. It's even a very small section. Most of the DNA in our genome isn't genes. It's mostly stuff that's in between genes. But you just need to learn that. That's one of those things you just got to learn. And it's quite surprising that 75% of students didn't know that because knowing what a gene is really quite important in biology. 50% of students couldn't describe one use of enzymes in the process of genetic engineering. There are 3. Removing the human gene from its host, isolating the gene, you use enzymes for that. Might be several enzymes involved in that process. You need to cut the plasmid open. Here it is, cutting the plasmid open. That requires an enzyme. It's called a restriction enzyme that cuts at a certain site. And then inserting the DNA into the plasmid requires another enzyme called DNA ligase. You don't need to know the names of those enzymes. You just need to know that enzymes are involved in those processes. An enzyme is not involved in putting the plasmid into the bacterial cell. And it doesn't really help that they have a bacterial cell here that looks like it's got a vacuole in it, for anybody who was doing the vacuole question. I think that's supposed to be the genomic chromosome of the bacterium, but it looks awfully like a vacuole, doesn't it? And that's not going to help anybody in that particular exam when they were asking about vacuoles. So bad SQA. Okay. Name another type of protein produced by the immune system in response to pathogens, and that's an antibody. A lot of people forget that an antibody is not a cell, it's a protein, and it's made by the cell, by the lymphocyte, and it's sent out into the blood to attack pathogens. So when you're writing your list of proteins to learn, make sure you include antibody in it. It's a kind of protein. I think the problem here that a lot of students missed out on was that they weren't clear that oxygen being present meant you were going to go on and do aerobic respiration, not fermentation. So a lot of students got the first part of this, they got the marks for saying, let's look at the diagram, they got the marks for saying glucose to pyruvate and 2 ATP, but they didn't carry on. And what happens is oxygen's taken into the mitochondrion, where the pyruvate's broken down to carbon dioxide and water, 36 ATP produced, and carbon dioxide sent off as waste. If you didn't recognize that the oxygen being present was required for aerobic respiration, which happens in the mitochondrion, you probably missed that. They didn't call it aerobic respiration, which is probably what everybody flags to. But it's just a question of knowing what aerobic respiration actually is. It's the respiration, the breakdown of pyruvate using oxygen. Okay? This looked like a simple reading of the question problem. 50% of the students missed this one because they were asked to name structure S. They just assumed they were going to be asked to be naming structure T. They hadn't read the question carefully. And I think a lot of students, according to the report, a lot of students just gave the name of the structure T, which is, of course, the medulla, instead of giving its function, which is what they were asked for. A simple question of just being careful there. And then state the role of receptors in the nervous system is to detect sensory input. This is a tricky old question because there are more than one type of receptor that we learn about. Because remember, we've got hormone receptors on the cell surface of cells in the liver that are detecting insulin and glucagon. So we learn about two different kinds of receptors. At the end of a sensory cell in the periphery, it has either a modification for detecting sensory input, which is called a receptor, or it's got a receptor body that might be, or even a receptor cell, a separate cell that does reception. And that passes the signal to the sensory neuron, which passes it to the spinal cord. But we do have this problem of, in the nervous system, you could probably answer that there are receptors in the synapse for chemical transmission because the neurotransmitter that comes across has to be taken in by a receptor. But that wasn't in the marking instructions, possibly because no students gave that answer, I think. It would be right, though. Okay. No problem here. This had lots of different possibilities for answering, so you got away with that, and there were no problems. Everybody put a version of that answer in that worked. The struggle with question C, though, which says 7C, name process B is mitosis. How do we know it's mitosis? Well, we've got a cell here. Cell A goes to this thing by process B, and what have we got? One cell going to 4 cells. Right? So the cell divides. We only learn one type of cell division in National 5, and that is mitosis. There are other types of cell division that we'll learn about later. Bacterial fission, meiosis. But the only one we have to write down here is mitosis. I don't think you got the mark for saying cell division. Maybe. They might have given it to you, but I don't think so. Mitosis is what you had to write down there. The problem here was 50% of students put gene here instead of allele. This is the versions of the gene are alleles. We're talking about all the same gene here, so we can't talk about these are both the same gene. And homozygous doesn't mean the same gene; it means the same allele of the gene. And heterozygous means two different alleles of the same gene. So it's clearer when you think about heterozygosity, so that you've got two different alleles of the same gene. Whereas here, it's the same alleles of the same gene, of one gene, right? It gets a bit confusing. You just need to be clear in your learning there. Flashcards are so important for this sort of stuff. This is a transpiration question, and transpiration is an experiment that we don't do very often in the lab because it's very hard to actually get it right. Very hard, and it takes time as well. It usually takes longer than a period, so we don't get to do it very often. And as a result, by not doing it physically, we often don't realize what these things are for. But the polythene bag, the purpose of this polythene bag stumped 75% of the students. And the purpose of it is to ensure that all the water loss of the plant leaves via transpiration. So what they did was they weighed them. So everything inside the bag, water vapor or whatever, was included in the weighing. Any water that left the system that wasn't weighed had to go through the leaves. That was the point, to ensure all water loss was through the leaves via transpiration, right? But again, I think that was just because we haven't done the experiment very often. It's not an easy experiment to carry out in the lab. Percentage change, and that gives us the reason for the percentage. The starting mass was different for each plant. That's all you have to say. Okay, it's called normalization. We're making sure that we can treat all of these plants the same way by looking at their percentage increase in mass rather than their absolute increase in mass, which is what is measured here. And with reference to the aim, give a conclusion for the investigation. Well, what you couldn't say, or what you shouldn't say, is that the lower leaf surface had 36% and the upper leaf surface at 4% or whatever. What you had to do was give it reference to the aim, and here was the aim. Look, investigation was carried out to find out which leaf surface loses most water through transpiration. Basically what you do is you rewrite this sentence with, replace the word which with upper or lower, and then so be lower in this particular case. Lower leaf surface loses most water through transpiration. You just, you don't restate the data, you rephrase the aim to show which result you got. So the lower leaf surface, okay? And I put it in here. Most water is lost through the lower surface of the leaves. This is a bit of a strange question, this one, but at least it was rather strange, some of their answer scheme stuff. I thought this answer was, we'll come to that, okay? 50% didn't give an explanation. They wrote down that the shape was a biconcave disc and got a mark for that, but didn't explain why it's important. The biconcave disc increases the surface area to volume ratio for efficient diffusion of oxygen, or increases the surface area is enough for the mark, I think. There were a couple of other possible options. No nucleus in the red blood cell. It means you can fit in more hemoglobin for transporting oxygen, or to fit through the capillaries, it makes the cell more elastic for fitting through capillaries. They also put this in the answer scheme, that biconcave was, the shape was to fit through the capillaries. That's technically not right. It's really more to do with the size. The capillaries are the same size as the cell, and the biconcave is a smooth, regular shape rather than a sickle, which is what gives you sickle cell anemia. So I don't know. They've kind of, biconcave is better than sickle cell, that's for sure, for fitting through capillaries, but it's more to do with its smoothness, the fact that they can't get tangled up with each other. But anyway, they give a mark for that, so well done if you if you got that mark, because I'm not sure you deserved it, but anyway, there you go. Again, the big problem here was not reading the question. You've got to read the questions. Hit yourself on the head on the morning and say, I've got to read the questions, okay? And make sure you read them carefully. And when you go back over your paper at the end, if you've got time, make sure you read them again, okay? Again here, a lot of students, 50% of the students talked about the function of the blood vessels, not their structure. And that's all you had to write there if you did a little table in your book. Any three of those four, in fact, walls, valves, and channel are the main ones. Cross-section is not usually mentioned. It wasn't in the answer scheme either, but it would get you a mark if you put it in. The wall of the artery is thick, etc. So that's what your structure only, not function. If you say that artery was to carry blood away from the heart, that's fine, but you're not going to get a mark for it, right? If the vein is carrying blood towards the heart, that's fine, but you're not going to get a mark for it. And if you say any of those wrong, you'll get marks taken away, probably. Saying anything wrong will negate marks. Okay, so this one caused a lot of problems too. Nearly all students, it says, so we'll say 90%, right? Couldn't tell the difference between validity and reliability. And they've been on a bit of a rift for this for about 3 years, the SQA, so make sure you know the difference between validity and reliability. Validity means that everything you can control is being controlled, or everything that's possible to control. The more things you control in the experiment, outside of your independent variable, which you're changing the value of, and your dependent variable, which you're measuring, the more valid your experiment, right? Validity is all about controlling your experiment. Reliability is about repeating your experiment, especially in biology, where different organisms have different properties. Or every individual is genetically different, of course. So reliability is about taking lots and lots of different measurements. Now, a lot of people put reliability in here instead of answering the question about validity. That says valid there. And make sure you know the difference between those two things. There's a definition in probably in your notes from your teacher. Make sure you read it and make sure you get it right. Okay? A lot of people are not getting it right. They're bound to ask it again this year. That's what I would think. No guarantees, though. Okay, what else happened here? No, everyone did okay on that one. Everyone did okay on question 12. Question 12. Looks good. Everyone did okay there. Looks like everyone did okay there. Question 13. Yep, all good. Question 14. This was a bit of a problem. I found this actually a difficult question because you have to see this word here, setting up. Right? I think about using a pitfall trap when they give you a question like this and identify two variables that should be kept constant when setting up this sampling technique. We're putting up several pitfall traps and then they all have to be the same size. They all have to have the same type of camouflage on them, that sort of thing. I read this question initially thinking, what do I have to be careful of when I come back to measure the trap? So measuring at the same time every day, that sort of thing. But that's not it. It's just setting up. So if you're going to put up several traps, which is what you have to do for reliability, and most people got that one okay with reliability, you've got to make sure they all look the same, right? The type of camouflage, the type of leaf and stuff on top is going to change the number of type of insect that comes in there, right? It's going to change it. So you've got to make sure they're all the same as much as possible, right? It's controlling the variables. Caused a lot of problems. The evolution question caused quite a few problems. There's a copyright picture of a wren. That is actually a valid picture of a wren in this case. Suggests how scientists could prove that the St Kilda wren is not yet a different species from the Scottish mainland wren. Well, you're the same species if you can breed with each other and produce fertile offspring. So they can test to see if they breed. And you can use they because the St Kilda's wren is the one mentioned. As long as you make sure one is mentioned here, they breed with mainland wrens to produce fertile offspring. That'll get you the mark. Half the students didn't get that mark. And here, the order of events in the formation of a new species is what you had to do here. I think B and E are probably the tricky ones. Most people realized, I think, that isolation is required, a barrier of some kind. So they didn't use the word barrier here, so that might have caused a few problems because the SQA National 5 guidance calls that a barrier rather than isolation, but isolation counts as a barrier. It's geographical barrier, right, because you come on an island. D, mutation, then you get mutation. Then you get natural selection, right, sorts out the mutations to see which are any good. Then you get over time. Time needs to happen. Each subpopulation, so we get genetic drift in the populations. Eventually, the gene pools are too different for the species to get back together and breed. Okay. I think this was a poor question. In the report, the SQA didn't see it as a poor question, but we were asked to read these questions carefully. So what they needed to leave out was evolution of species, right? Because here's the answer they wanted. Best adapted individuals survive to reproduce. Favorable alleles are passed on to offspring, right? That's not a definition of evolution of species. That's a definition of natural selection. So they asked about the evolution of species, but they were only asking about natural selection. So it's possible that a lot of students tried to answer too much there or try to cover all these bases, when in fact the only thing they were being asked about was explain natural selection. That's what that answer is. That's the answer they wanted. Okay? I'm not sure it was a very fair question, to be honest. Sometimes that happens. This one caused a lot of problems. A lot of students, 90% of students didn't get this. So I would say a lot, we'll say 90%, more than 75% anyway. And the problem was they saw this 5%, they saw that number there, plant plankton, they saw that. So they worked out what 5% of 42,000 was, right? And stuck it in there. And that wasn't the answer. Because it's not 5% of 42,000. 42,000 was 5% of the total amount of light, and we had to calculate what the total amount of light was. So 42,000 was the 5%. Turns out the answer is 100% would be 840,000. How do you work it out? Well, there's three ways you can work it out. You can use the fractional method. So 5% is 1/20th. So you can multiply your 42,000 by 20 and you'll get that. Or you just remember a factorial thing. So 20 times 5, it gets you to the 100%, which is your total. Gets you the same thing. Or you can do it through percentages. 42,000 is the value for 5% of the total, right? 5 over 100 of the total. There you go. And that's it, I think, in the end. You had a lot on there, and the main thing to do is relax, take it calmly, and just do the best you can. And remember all this stuff, right? If you're asked to describe, put in a full statement. If you're asked to explain, give a reason. If you're asked to compare, you must talk about the similarities and or differences depending on what you're being asked. Good luck in the exam. Hope you all do well.